Imagine a world where your body's most elite defense force, the very system designed to protect you from every microscopic threat, is systematically dismantled from within. This isn't a dystopian novel, it's the chilling reality of human immunodeficiency virus, or HIV. For decades, this insidious pathogen has waged a silent, relentless war against humanity, not by directly causing illness, but by meticulously destroying our ability to fight off disease. It's a master of biological sabotage, turning our own cellular machinery against us. Today, we're going on an unprecedented journey deep into the microscopic battlefield to uncover exactly how HIV launches its devastating assault and ultimately cripples the human immune system, transforming a robust defense into a house of cards vulnerable to the slightest breeze. Before we dive into the destructive mechanisms, let's establish what HIV actually is. HIV is a retrovirus, a unique class of viruses that carry their genetic information in the form of RNA rather than DNA. This distinction is crucial because retroviruses possess a remarkable enzyme called reverse transcriptase, which allows them to convert their RNA into DNA. This newly synthesized viral DNA can then be integrated directly into the host cell's genome, essentially rewriting the host's genetic blueprint. This integration is a cornerstone of HIV's persistence and its ability to evade immune detection for extended periods, making it a particularly challenging adversary. The journey of HIV within the human body begins with its entry. Unlike many viruses, HIV doesn't just indiscriminately infect any cell it encounters. It has a specific target, the CD4 plus T lymphocytes, often simply called CD4 cells. These cells are the orchestrators of our adaptive immune response, acting as crucial communicators and activators for other immune cells. Think of them as the generals and strategists of your body's defense army. HIV uses specific proteins on its surface, primarily GP120 and GP41, to bind to the CD4 receptor and a coreceptor, either CCR5 or CXCR4, on the surface of these T cells, initiating the fusion process that allows the virus to enter. Once inside the host cell, HIV begins its insidious work. The viral RNA, along with the reverse transcriptase enzyme, is released into the cell's cytoplasm. This is where the magic, or rather the menace, of reverse transcriptase occurs. It meticulously transcribes the single-stranded viral RNA into a double-stranded DNA copy. This step is a critical vulnerability point for the virus, and many early antiviral drugs were designed to specifically inhibit reverse transcriptase, blocking this crucial early stage of viral replication and preventing the virus from embedding itself into the host's genetic material, thereby slowing down the progression of the disease. With the viral DNA now synthesized, the next critical step is integration. The newly formed viral DNA travels into the cell's nucleus, where another viral enzyme, integrase, plays its role. Integrase precisely cuts the host cell's chromosomal DNA and inserts the viral DNA, now called a provirus, directly into it. This is a point of no return for the infected cell. Once integrated, the provirus becomes a permanent part of the host cell's genetic material, silently residing there, capable of remaining dormant for extended periods or activating to produce new virus particles. This integrated provirus is why HIV infection is lifelong. After integration, the provirus can enter a state of latency, remaining dormant for months or even years. This latency phase is one of the biggest challenges in eradicating HIV, as the integrated viral DNA is essentially hidden from the immune system and most antiviral therapies. However, when the host cell becomes activated, for example, during a normal immune response to another infection, the provirus can also become activated. This activation triggers the host cell's machinery to transcribe the viral DNA back into viral RNA, initiating the production of new viral components, effectively turning the cell into a virus factory. The viral RNA produced during activation serves multiple purposes. Some of it acts as messenger RNA, mRNA, to direct the synthesis of viral proteins, while other strands become the genetic material for new virus particles. The host cell's ribosomes and other cellular machinery, originally meant for producing its own proteins, are hijacked to create long chains of viral proteins. These precursor proteins are non-functional on their own and need further processing. This hijacking process places immense stress on the infected cell, diverting its resources and ultimately contributing to its demise as it is forced to prioritize viral production. To become functional, the long chains of viral proteins must be cut into smaller, individual proteins. This crucial task is performed by another HIV-specific enzyme called protease. Protease cleaves the precursor polyproteins into distinct structural proteins, like capsid proteins, and enzymatic proteins, like reverse transcriptase, integrase, and protease itself. Without this precise cleavage, the new virus particles would be immature and non-infectious. Protease inhibitors, another class of highly effective antiretroviral drugs, target this enzyme, preventing the maturation of new virions and thus halting the spread of the virus. Once all the viral components, genetic material, RNA, enzymes, reverse transcriptase, integrase, protease, and structural proteins, have been synthesized and processed, they assemble near the inner surface of the host cell's membrane. This assembly is a highly organized process, with the viral RNA and enzymes encapsulating within the newly formed capsid proteins. The viral core then buds off from the host cell, acquiring a piece of the host cell's membrane as its outer envelope. This budding process often damages the host cell membrane, further contributing to its eventual destruction, releasing a multitude of new, infectious virions. Each infected CD4 plus T cell can produce hundreds to thousands of new HIV particles before it eventually dies. These newly formed virions are then released into the bloodstream, ready to infect other healthy CD4 plus T cells, perpetuating the cycle of infection. This exponential replication is what drives the rapid depletion of CD4 cells, especially in the early stages of infection. The sheer volume of viral production overwhelms the immune system's ability to replace these vital cells, leading to a gradual but steady decline in immune function over time, setting the stage for more severe complications. The primary target of HIV, the CD4 plus T cell, is not just any immune cell. It's a central figure in orchestrating both cellular and humoral immune responses. Helper T cells, as they are often called, release cytokines, signaling molecules that activate B cells to produce antibodies, cytotoxic T lymphocytes, CTLs, to kill infected cells, and macrophages to engulf pathogens. By systematically destroying these critical cells, HIV effectively cripples the entire immune system's ability to recognize and fight off a wide range of infections and even certain cancers. It's like removing the command center from a military operation. The destruction of CD4 plus T cells occurs through several mechanisms. Firstly, the massive replication of HIV within the cell often leads to cellular exhaustion and programmed cell death, or apoptosis. Secondly, the budding of new virions from the cell surface can physically damage the cell membrane, compromising its integrity. Thirdly, uninfected CD4 cells can sometimes fuse with infected cells, forming giant syncytia that are dysfunctional and eventually die. Lastly, even uninfected CD4 cells can be targeted and killed by the immune system itself, if they display viral proteins on their surface or are mistakenly identified as infected, a phenomenon known as bystander killing. The clinical progression of HIV infection can be broadly divided into 3 stages. The first is the acute infection phase, which occurs a few weeks after initial exposure. During this stage, the virus replicates rapidly, leading to a sharp drop in CD4 cell counts and very high viral loads. Many individuals experience flu-like symptoms, such as fever, fatigue, rash, and swollen lymph nodes. This phase is characterized by an intense but often unsuccessful attempt by the immune system to control the burgeoning viral population, with a significant battle being waged in the lymph nodes. Following the acute phase, most individuals enter a prolonged clinical latency stage. During this period, which can last for many years, even a decade or more without treatment, HIV continues to replicate, but at a lower level. CD4 cell counts gradually decline, but the body's ability to regenerate these cells often keeps pace for a while, maintaining a relatively stable, albeit slowly diminishing, immune function. People in this stage may experience no symptoms or only mild, non-specific ones like persistent swollen lymph nodes. This silent period can be deceptive, as the virus is still actively destroying immune cells. The third and final stage of HIV infection, without effective treatment, is acquired immunodeficiency syndrome, or AIDS. This diagnosis is made when an individual's CD4 cell count drops below a critical threshold, typically 200 cells per cubic millimeter of blood, or when they develop one or more specific opportunistic infections or certain cancers that are characteristic of severe immunodeficiency. By this point, the immune system is profoundly compromised, leaving the body extremely vulnerable to pathogens that a healthy immune system would easily fend off, leading to severe illness and ultimately, death. Opportunistic infections are the hallmark of AIDS. These are infections caused by pathogens, bacteria, viruses, fungi, parasites, that typically do not cause disease in people with healthy immune systems but seize the opportunity to attack a weakened one. Examples include pneumocystis pneumonia, PCP, Kaposi's sarcoma, toxoplasmosis, cryptosporidiosis, and various fungal infections like candidiasis. These infections are often severe, widespread, and difficult to treat, as the body lacks the crucial T-cell-mediated immunity to mount an effective defense against them, leading to debilitating symptoms and organ damage. The long-term depletion of CD4 plus T cells has far-reaching consequences beyond just opportunistic infections. It also impairs the immune system's ability to control certain cancers, such as Kaposi's sarcoma, non-Hodgkin lymphoma, and cervical cancer, which are more common and aggressive in individuals with AIDS. The chronic inflammation induced by persistent viral replication also contributes to various non-AIDS-defining conditions, including cardiovascular disease, kidney disease, and neurocognitive disorders, highlighting the systemic impact of unchecked HIV infection on almost every organ system in the body. The impact of HIV isn't confined to CD4 plus T cells. Other immune cells, such as macrophages and dendritic cells, can also become infected. While these cells are less prone to cell death upon infection, they can serve as viral reservoirs, silently harboring the virus and contributing to its persistence. Macrophages, in particular, can transport the virus to different parts of the body, including the brain, contributing to HIV-associated neurocognitive disorders, HAND. Dendritic cells, normally crucial for initiating immune responses, can also become vehicles for viral dissemination, carrying HIV to lymph nodes where it can infect more T cells. The gut-associated lymphoid tissue, or GALT, is an early and significant site of HIV replication and CD4 plus T cell depletion. The GALT contains a vast number of CD4 plus T cells and is essentially the largest immunological organ in the body. Within days of infection, HIV rapidly depletes CD4 cells in the gut, leading to a breakdown of the gut mucosal barrier. This allows bacteria and their products from the gut lumen to translocate into the bloodstream, triggering chronic systemic immune activation and inflammation, even in the absence of high viral loads, further stressing the immune system and contributing to disease progression. Chronic immune activation and inflammation are critical drivers of HIV pathogenesis, even when viral load is suppressed by treatment. The constant stimulation of the immune system, attempting to fight a persistent infection, leads to a state of exhaustion and dysfunction. This chronic activation accelerates the turnover of immune cells, including uninfected CD4 cells, making them more susceptible to infection and apoptosis. It also contributes to generalized inflammation throughout the body, linking HIV to non-AIDS defining conditions like cardiovascular disease, kidney disease, and accelerated aging, making it a truly systemic illness. One of the most challenging aspects of HIV is its ability to mutate rapidly. Reverse transcriptase is prone to errors, leading to frequent changes in the viral genome. These mutations can alter the proteins on the viral surface, allowing HIV to escape detection by the immune system's antibodies and cytotoxic T lymphocytes. Furthermore, mutations can lead to drug resistance, making existing antiretroviral therapies less effective. This high mutation rate is why combination antiretroviral therapy, ART, is so crucial, as it targets multiple viral processes simultaneously, making it much harder for the virus to develop resistance to all drugs at once. The discovery of antiretroviral therapy, or ART, revolutionized the fight against HIV. ART involves a combination of several drugs from different classes, each targeting a specific stage of the HIV life cycle. For instance, nucleoside reverse transcriptase inhibitors, NRTIs, and non-nucleoside reverse transcriptase inhibitors, NNRTIs, block reverse transcriptase, protease inhibitors, PIs, prevent viral protein maturation, integrase inhibitors, INSTIs, block integration, and entry inhibitors block the virus from entering cells. This multi-pronged attack dramatically reduces viral load and allows the immune system to recover, significantly extending life expectancy. The success of ART has been nothing short of miraculous. For individuals who adhere to their treatment regimen, ART can suppress the viral load to undetectable levels. This not only prevents progression to AIDS but also makes the person effectively non-infectious, a concept known as undetectable equals untransmittable or U equals U. This profound scientific breakthrough has transformed HIV from a fatal diagnosis into a manageable chronic condition, allowing people living with HIV to lead long, healthy, and fulfilling lives, while also dramatically reducing new transmissions. Despite the remarkable advancements in treatment, HIV eradication remains a formidable challenge. The main reason is the existence of viral reservoirs. These are groups of infected cells, primarily resting memory CD4 plus T cells, that harbor integrated provirus in a latent state. These cells do not actively produce new virus particles, making them invisible to the immune system and unaffected by current ART drugs, which only target actively replicating virus. When ART is stopped, these latent reservoirs can reactivate, leading to viral rebound and a resurgence of the infection, necessitating lifelong treatment. Research into finding a cure for HIV primarily focuses on two main strategies: shock and kill and block and lock. The shock and kill approach aims to activate these latent viral reservoirs, forcing the cells to produce virus, thereby making them visible to the immune system or susceptible to ART-induced cell death. The block and lock strategy, conversely, seeks to permanently silence the latent provirus, preventing it from ever reactivating. Both approaches face significant hurdles, but ongoing research offers hope for a future without the need for lifelong ART. Beyond treatment and cure, prevention strategies are equally vital. Pre-exposure prophylaxis, PrEP, and post-exposure prophylaxis, PEP, are revolutionary tools. PrEP involves HIV-negative individuals taking antiretroviral medication daily to significantly reduce their risk of acquiring HIV if exposed. PEP involves taking ART within 72 hours of potential exposure to prevent the virus from establishing a permanent infection. These pharmacological interventions, combined with consistent condom use, harm reduction strategies for drug users, and early diagnosis, form a comprehensive prevention toolkit. The global impact of HIV and AIDS cannot be overstated. Since the beginning of the epidemic, tens of millions of lives have been lost, and countless communities devastated. While significant progress has been made, particularly in access to treatment in many regions, millions still live with HIV, and new infections continue to occur daily. Disparities in access to testing, treatment, and prevention services persist, particularly in low-income countries and among marginalized populations, highlighting the ongoing need for sustained global efforts. Stigma and discrimination remain major barriers in the fight against HIV. Fear and misinformation often lead to social ostracism, preventing individuals from seeking testing, treatment, and support. This not only harms individuals but also undermines public health efforts to control the epidemic. Education, empathy, and challenging misconceptions are crucial to fostering an environment where people living with HIV can live openly and access the care they need without fear of judgment. The human aspect of this virus is as complex as its biology. Looking forward, the scientific community continues to push the boundaries of research. Efforts are underway to develop an effective HIV vaccine, which would be a game-changer in preventing new infections globally. While progress has been slow due to the virus's unique characteristics and variability, new approaches and technologies are being explored. Additionally, long-acting injectable forms of ART are being developed, which could improve adherence and simplify treatment regimens, making it easier for people to manage their condition. In conclusion, HIV is a retrovirus that primarily targets and destroys CD4 plus T cells, the orchestrators of the adaptive immune response. Its life cycle involves entry, reverse transcription, integration into the host genome, protein synthesis, assembly, and budding, ultimately leading to the progressive depletion of immune cells. This systematic destruction culminates in AIDS, leaving the body vulnerable to opportunistic infections and cancers. Understanding this intricate attack is key to appreciating the complexity of the virus and the ingenuity of the scientific community's response. The journey of understanding HIV has been one of both immense challenge and incredible scientific triumph. From the early days of a mysterious, fatal illness to the present era of effective treatments and robust prevention strategies, humanity's resilience and scientific prowess have been put to the test. Yet, the fight is far from over. Millions still live with the virus, and the quest for a definitive cure and a preventive vaccine continues with unwavering dedication. It's important to remember that HIV is a virus that knows no boundaries of geography, race, gender, or sexual orientation. Its impact is a global one, and therefore, the response must also be global. International collaboration, shared resources, and equitable access to healthcare are paramount in reaching the goal of ending the HIV epidemic. Every individual's effort, from advocating for awareness to supporting research, contributes to this collective endeavor, ensuring that progress continues and no one is left behind in the fight against HIV. The story of HIV also serves as a powerful reminder of the incredible complexity and delicacy of our own bodies. The immune system, a marvel of biological engineering, is a testament to evolution's ingenuity. Yet, even this sophisticated defense can be brought to its knees by a tiny, adaptable pathogen. This ongoing battle underscores the importance of continued scientific inquiry, not just for HIV, but for understanding and combating all emerging and re-emerging infectious diseases that challenge human health. Moreover, the lessons learned from HIV research have had profound ripple effects across many other fields of medicine and immunology. The development of ART paved the way for new drug development strategies. Our understanding of viral latency, immune evasion, and chronic inflammation has been significantly advanced. Even vaccine development technologies, including mRNA vaccines, have benefited from foundational research initially conducted in the context of HIV, demonstrating how challenges can drive innovation and lead to unforeseen benefits for humanity. As we conclude our deep dive into how HIV attacks the immune system, take a moment to reflect on the immense progress made and the challenges that still lie ahead. The intricate dance between host and pathogen, the relentless scientific pursuit, and the human stories behind every statistic all paint a picture of a global health crisis that has profoundly shaped our modern world. Your understanding of this virus is a step towards a more informed and empathetic world. Remember, knowledge is power. By understanding how HIV works, we can better appreciate the importance of prevention, treatment adherence, and the tireless work of scientists, healthcare providers, and advocates. Share this knowledge, challenge stigma, and support efforts to achieve an AIDS-free generation. The war within might be invisible, but our collective response must be clear, visible, and unwavering. Thank you for joining us on this detailed exploration of HIV's impact on the immune system. If you found this information valuable, please consider liking this video, subscribing to our channel for more in-depth scientific content, and sharing it with others. Your engagement helps us continue to produce informative and impactful videos that shed light on critical health topics. Stay curious, stay informed, and together, let's continue to learn and grow.